Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other. So here we will discuss about the lipoprotein and their lipoprotein contain apoprotein. And we will discuss about the lipoprotein containing apoprotein physiology and effect at concentration of triglycerides. And digestion of fat, obviously will discuss here. So first of all, we will understand the content of apoprotein on the lipoprotein in lipoprotein. So the lipoprotein is made up of phospholipids, unilayer, and apoprotein as well as triglyceride and cholesterol. So here is the micelle, which that is without protein, apoprotein, while that is the fat is diglyceride and monoglyceride and phospholipids. On the other hand, the very low density lipoprotein is also known as chylomicron, containing ApoB48 protein. On the other hand, the IDL, which contain a less amount of protein, ApoE and ApoB100, which that is the very ideal. So the IDL contain a ApoB100. And next, when LDL, which that is known as low density lipoprotein, contain a slightly more protein and ApoE contain. On the other hand, HDL contain a ApoE, ApoC2 and ApoE also. So on the basis of these, protein is high and cholesterol is low, as well as triglyceride is low in the high density lipoprotein. So the high density lipoprotein is good, but low density lipoprotein is very bad. But in this video we will discuss all the mechanism, and we will discuss and cover the whole concept about the lipoprotein. So here is the digestive system, which that's contained by gallbladder, and bile duct contain and that's produce bile acids, cholic acid, as well as bile salts, cholesterol. So here is the pancreas, which produce the pancreatic lipase enzyme, which that both combine, and these combine due to the CCK, and CCK is used to contract the bile gallbladder due to the effect of the chyme which that are present in the duodenum, which that is the fat. So this effect is contract due to the enteroendocrine cell, which give the signal to the gallbladder. So important to know that here is the duodenum. So the duodenum contain enterocyte, is also known as brush border cell, which contain enzyme. But enterocyte is also contain the whole other cell is known as enteroendocrine cells. But here we will not discuss about the enteroendocrine cell, but first just understand the digestion of fat and transport in the form of lipoprotein. So here's the duodenum, which contain enterocyte is here. So this enterocyte is very important function, which that absorb micelle, which come from the dietary fat, and this micelle absorb into the enterocyte. So here is the blood vessel. First of all go the lipoprotein fat into the lacteal, which that is a lymphatic system. But here we will not understand, just make a concept. First go into the lymphatic system. So here is the fat, which that is the dietary fat. How it's digest by the to convert into micelle. So that's bile acid, cholic acid, bile salt, and cholesterol, which that is used for the emulsification of fat. So this emulsification is due to the produce amphiphilic micelle, which that is a both side is hydrophilic. So that is why it's known as amphiphilic. So the pancreatic lipase enzyme used to produce micelle. So here is a contraction by the CCK due to the enteroendocrine cell produce give the signal. So endocrine system. Here is the micelle is formed. So micelle go into the enterocyte. So first of all we will discuss into the dude enough for understand that is go into the enterocyte. So uh, basically basically np npc npcil1, which that is enzyme used to convert the very low density lipoproteins known as chylomicron, which contain apoB48. But here is the inhibitor which drug use. Its name is azitimibal, which that is used for the inhibition of micelle. So that's micelle will production inhibition for the chylomicron production. So here is the ApoB48 will be added in the micelle to produce very low density lipoprotein, which contain a high number of fat but the low number of protein as well as low number of the cholesterol, which that is the come from the dietary cholesterol, not synthesized into the liver. So next we will discuss about the liver synthesis of cholesterol. So first of all we should understand that is the chylomicron go into the enterocyte. First of all, it's formed in the enterocyte. So remember, so here is the bile acid production. If we can prevent the bile acid production from this drug is known as choleservatilum, which that is inhibit and this drug is inhibited by this bile acid to stop the emulsification of fat. So here this bile acid can also go into the blood. So these bile go into the blood also. That's bile acid go into the liver to recycle. So this recycle is again go into the gallbladder for excretion. So the 48 ApoB is also enter inside if basically if not absorbed. So the bile acid release and this release bile acid go into the hepatocyte. But remember that it's not necessary to remember these things or can get concept. So here we will discuss about the when enterocyte will release the VLDL chylomicron into the blood vessel. So it's come from lacteal, so the lymphatic system, and VLDL when give the basically VLDL when give the uh, fat transportation into the adipose tissue and other tissue to decrease their fat. So here is the enterocyte. First we will understand about the mechanism of enterocyte, the micelle conversion into the chylomicron. So the chylomicron, here is the lumen and here is the micelle which that is entered into the enterocyte, and the triglyceride, cholesterol and ApoB48 protein is added in it to produce very low density lipoprotein as a chylomicron. So this chylomicron is transported into the lymph, and that's after all that's transport into the blood vessel. So the blood vessel is a very low density lipoprotein containing. Here that is converted into the IDL when transport the fatty acid and triglyceride to the adipose tissue and peripheral tissue, all peripheral tissue, as well as where the need of the triglyceride and fat they can transport. So here is a very low density lipoprotein which that is give the fatty acid triglyceride by the to the adipose tissue by the receptor is known as low density lipoprotein receptor 1, which that is give by the ApoE, that is apo E side when can get the ApoE protein. So that's a very low density lipoproteins contain ApoB. So here is the ApoB is used for it. So remember, on the other hand, a low density lipoprotein receptor is also present in the adipose in the peripheral tissue. For example, muscle and for example smooth muscle and cardiac muscle or skeletal muscle. That's all thing. So when transport these fats and other part of fats and triglyceride when give that to the tissue and adipose tissue, so that will be convert and that produce the IDL. So this IDL is the basically uh, not a IDL, but it's the remnant chylomicron. So we can give the name is known as the chylomicron remnant by the lipoprotein lipase enzyme used to it. So the remnant chylomicron can get the protein from the endothelial cell of the blood vessel to receive the ApoE site. So the ApoE protein is also added in it. And this ApoE is used for the chylomicron remnant receptor in the hepatocyte. So here is the hepatocyte which contain a chylomicron remnant receptor, or also known as low density lipoprotein receptor, LDL receptor basically. So when enter this, the receptor contain a vesicle which that's endocytosed. It is known as endocytosis, and the type of endocytosis is known as the phagocytosis because the solid particle. So that is why it's not liquid. So the endosome will be formed. Endosome will separate the receptor, and endolysosome will be formed by the lysosome add in it. So that's very low density lipoprotein endosome form, and this is the endolysosome lysomine splitting. So these split into the triglyceride, cholesterol, fats, phospholipids, and many more. So these triglyceride, phospholipid, cholesterol is again convert into the very low density lipoprotein. So how it's possible? Let's talk. Here is the high HMG CoA reductase enzyme used to convert the acetyl CoA to cholesterol. So the here is the inhibitor is the statin which used to inhibit the production of cholesterol. So this inhibition can lower the cholesterol, very low density lipoprotein and cholesterol. So this inhibitor is the lowering agent of drug, which that's lower the cholesterol formation. So here is the another thing is the MTP is the microsomal triglyceride transfer protein is used to transfer the apoprotein E, ApoC, and Apo 100, which that is convert a very low density lipoprotein. So the MTP inhibitor is the lomit lomit lomiti lomiti lomit lomitapide. So the lomitapide is the drug which can inhibit the MTP to prevent the production of low density lipoprotein, LDL. So the LDL will decrease. So the LDL property contain a ApoE, ApoC, Apo 100, which are used for the giving the adipose tissue fat to the adipose tissue and peripheral tissue. On the other hand, it's also give to the cholesterol to the plasma membrane by the ApoC. So here is a basically we can say that is very low density lipoprotein can give the multiple region to give the fat and cholesterol. Adipose tissue receive the fat in the form of triglyceride, and adipose other peripheral tissue, for example skeletal muscle, which receive the fatty acid. On the other hand, plasma membrane is also receive the cholesterol and phospholipid, which that is the single layer of the LDL, very low density lipoprotein contain, and that's uh, give the phospholipids also for synthesis of plasma membrane of the cell membrane. So the plasma membrane. So here I am drawing that the ApoE site, which give the which is bind with the LDL receptor of the adipose tissue. to give the triglyceride. And that is for example fatty acid, which that is also convert the triglyceride in the adipose tissue by the here is the wall of the blood vessel contain a lipoprotein lipase enzyme which use for it. So the lipoprotein lipase enzyme used to give the triglyceride. On the other hand, here the free fatty acid is also received by the tissue, and that's converted into the ApoB100 and ApoE side, and the ApoE side, if one side will be lost, and the lipoprotein lipase enzyme used it to produce these things. On the other hand, there's ApoE and ApoB100 contain IDL, intermediate density lipoprotein, which means the 2 unit of lipoprotein contain here, and that can be moved back by the LDL receptor to endocytose or scavenger receptor. It's also known as LDL receptor is also known as scavenger receptor, which that receive the IDL to again recycle, and that's cholesterol picking property, which that's pick the cholesterol from the tissue to give back into the liver, and that liver will be stored into the bile and excrete. So remember, on the other hand, here is the low density lipoprotein will be formed. Low density lipoprotein contain ApoB100 just. So the ApoB100 just received by the receptor is known as PCSK9. So the PCSK9 inhibitor, which that will not allowing to enter the low density lipoprotein is the alirocumab. So this drug, and here is the hepatocyte are present. For example, that's other is the also hepatocyte, which that is in the form of liver structure, but here is for understanding that is the here is the low density lipoprotein LDL receptor ApoB100, which that receive the cholesterol, which that receive the cholesterol, and that's bind high density or low density lipoprotein. Low density lipoprotein bind with the hepatocyte to go inside is known as endosome. So this endosome form is the endolysosome, and this endolysosome will be split. Lysis will be occur to produce a more thing, triglyceride, cholesterol, and other phospholipids, which that is recycled again. So that is the inhibitor for receiving the low density lipoprotein. Why? Because inhibition is very important when the low density lipoprotein deficiency in our blood. So when in our blood will be low deficient, or on the other hand, if the fatty liver will be produced because the high amount of cholesterol and fat are present in the liver, so this inhibitor can be work well. So here I am drawing that is the low density lipoprotein can affect the endothelial cell of the blood vessel. It LDL can enter into the endothelium. So here is the tunica intima. So tunica intima is the gap. So this gap is between the endothelial cell and smooth muscle cell of the tunica media. So the tunica intima is the middle, which that is receive the LDL when the endothelial cell dysfunction or some other major problem, which that is the high level of low density lipoprotein, which contain ApoB100. So here is the lumen, which contain a blood vessel. So that is why LDL move inside by the dysfunction of endothelial. Or the other problem is that uh, hyperlipidemia is other disease which can create these things. So this LDL will go enter and become oxidized, and this oxidized produce reactive oxygen species, and this reactive oxygen species which stimulate the endothelial cell, and this endothelial cell will stimulate the monocyte to enter, which that is the part of the immune cell, and these monocytes will enter to become a proliferate, and these proliferate to produce the macrophage. And this macrophage is very important character is that scavenger receptor is present in the upper surface of the macrophage, which that developed after the proliferation, and that's LDL will be received, and this LDL is the ApoB100 site to receive to enter inside to produce a foam cell. This foam cell is a cholesterol containing foamy formation, which that is a crystallized formation. After the crystal formation of the foam cell will be die and crystallization will be occur, and that is known as the plaque development, which that is the arteriosclerosis. So these arteriosclerosis become a thrombosis when thrombus formation during the rupturing of the endothelial cell of the blood vessel. So here is the tunica media, and tunica externa is also known as well. So let's talk about the very low density lipoprotein, intermediate density lipoprotein, low density lipoprotein, and high density lipoprotein, which that is the formation and how to become a less amount of cholesterol and high amount of triglyceride and their direction. So first of all, we will discuss here is the hypercholesteremia and hyperlipidemia and endothelial dysfunction or hyperglycemia, which that is a high level of sugar. High level of sugar is can cause the endothelial dysfunction because endothelial is not contain a insulin receptor, which that is receive the sugar without a receptor of insulin. So that's why too much sugar can affect and produce reactive oxygen species and more ATP. So here we will discuss about the amount of triglyceride and cholesterol with the different manner. So here is the ApoB48. So ApoB48 contain very low density lipoprotein. So this very low density lipoprotein contain triglycerides more, apoprotein B is more, while the cholesterol is low because it is not picking the cholesterol from the peripheral tissue, and it's ultimately converted by the giving the triglyceride to the cell to convert the ApoB100 and ApoE containing IDL. IDL mean intermediate density lipoprotein. So here the cholesterol will be decreased downside. So the decreasing of triglyceride is increased upside. It means the very low density lipoprotein contain more triglyceride but less amount of the cholesterol. For example, here I can draw here. That means when cholesterol will picking the cholesterol from the plasma membrane to become a filled high density lipoprotein. So the high density lipoprotein contain more cholesterol as compared to the very low density lipoprotein. But on the other hand, triglyceride is low in the high density lipoprotein field, but very much VLDL contain triglyceride. So all this video is about the lipoprotein physiology and their effect and how to inhibit the lipoprotein production and low density lipoprotein production to inhibition by the drug, as well as we are just understanding the physiology of this video. So thanks, bye.